Hi. So back to science. I'm a professor of biological sciences here at UCSD, and I'm kind of I don't know, a tinkerer. We like to get into problems where we think we can make a difference. But I'll just start by telling you that DNA, other than air, water, food, is your most precious possession. Each one of your cells has about six feet of this molecule, and it fits in about a thousandth of an inch. And you have about 40 trillion cells in your body, and each one of them have this DNA. And what this DNA is, and probably most of you know, is a recipe for how to make tiny, tiny machines inside your body that do everything and make you who you are. And there's thousands of these kinds of different proteins, and all of these recipes are inside your DNA. And sometimes a single point mutation, so like a single base wrong in your DNA, and it will give you horrible diseases. It could be Parkinson's disease, or make you look like an 80-year-old person when you're 8 years old. That's called progeria. And all of these are errors that happen in these macromolecular machines. And these macromolecular machines, proteins are like us. No sound, please. They actually build very sophisticated networks. They're very social. They do one thing in the morning and then bind to another thing. You can see they build networks and things look like cities. They actually come apart and come together, different kinds, and they do that at different parts of the cycle, of your cell cycle. Your cells divide and stuff like that. And so imagine that you have all of these machines working all at the same time at speeds that are amazing, like thousands of times these speeds, building highways and putting them apart. So imagine that we had a city where a highway just materializes right when you need it, and then it just falls apart afterwards because you just have to use it somewhere else. We bring molecules from one part of the cell to the other. Everything is chemistry. Everything is done by these machines that sort of like bring chemicals around. So very, very complex, very, very exciting, and all of this is coded in your DNA. And so these machines live inside cells. I told you you have about 40 billion of these cells in your body. And that's a cell, that's actually a human cell, where we've lit with light a particular molecule that's called actin. It's one of the highways that you saw in the previous movie. So you can see that this looks like at a city level, right? Like if you were sort of like had lights in the city, and that's what it looks like. And if we moved in, and so this is the realm of cell biology. This is the kind of images that people who study cell biology look like. Now, if you want to do a 10,000 zoom in, you would see one of those little teeny lines in there is a molecule that's made of atoms that was encoded in your DNA. And inside it, you can see chemicals, right? And so this is exactly, these machines do chemical reactions, and that's how everything in your body works. So all of these scales from the atoms all the way to your entire body define who you are and your biology and how long you're going to live. So obviously, we want to learn everything in all of these scales. Now, typically, well, we want to look at things at very high resolution. And so like look at all these chemical bonds and exactly how it works. How do you, process ATP? How do you have energy? How do your mitochondria become a powerhouse of the cell? We call this structural biology. We rip these molecules from the natural environment with biochemistry. We purify them, and then we can study them in a very, very acute detail. But now it's like you see someone in a freezer, right? You're not seeing a person interacting and interacting with their everyday life. In the other side is the realm of cell biology. That's cell biology over there, where what you want to do is study all of the connections. The difference between biochemistry and cell biology is organization. How do they come together? How do they act? When they act, the timing and the space of where they act. And the technique that I develop in my lab is called cryo-electron tomography, where we're trying to bridge these two. We're trying to look at these macromolecules in the exquisite biochemical detail where we can see where a mutation went wrong in your DNA, but really look at how then the whole cell goes wrong, and then how the whole tissue goes wrong, and what really leads you to disease. Now, I had the sort of privilege to get into this technique early. This got the Nobel Prize in 2017. The gentleman in the center is one of my mentors, Joachim Frank, where they developed this technology to look at these molecules in isolation. And that's great. But I didn't think it was enough. If electron microscopy is right now the unique and preferred technique to look at structures of biomolecules, why can't we just make it the best resolution imaging tool for cell biology? And so that's what we do, and we call it cryo-electron tomography. You'll see it's a little bit different. So I'm going to show you a movie. These are viruses. You can imagine it would be the same thing with cells. These are herpes viruses. We begin with the viruses floating around at room temperature or body temperature, and then we flash freeze them very fast with liquid nitrogen, like the tank some of you saw today, at liquid nitrogen temperatures, so fast that the water doesn't even have time to form crystals. So they remain almost like if it was liquid water. No structures are altered. And we can put this in a massive electron microscope. And then we basically take the sample and tilt it to different orientations, and we take pictures in different angles. This is exactly the same as a CAT scan, except that a CAT scan uses X-rays, and their samples are about your size, and this is 1,000,000 times smaller, or 1,000,000,000 times smaller. So we take pictures in different directions. For those of you who like signals, this is corresponding to one plane in Fourier space. So we do some mathematical operations in Fourier space, in 3D, and then we come back to real space, and this is all done automatically by computers. These microscopes, by the way, are multi-million-dollar microscopes connected to the data storage here at UCSD, so it's fantastic. And what we get are basically these gray value pictures that basically represent the structure. Literally, each pixel tells you about the atomic number of the atom that is in that place. And we can annotate this. And so this is a structure of a herpes virus that attacks humans. So this is the kind of work that we do in my lab. And this would have been all great and exciting, except we wanted to do more. And it turns out that cells, or like viruses or little bacteria or things like that, are a little bit too thick for the electron microscopes that we use. You have to make them very skinny to be able to look at them and just ridiculously high resolutions where we literally can do that movie that I showed you at the beginning, but like in real life. And so we freeze the cells. We can use fluorescent microscopy, which is the preferred method of cell biologists to look at things. But then we decided to steal a technology from material sciences. And this is when I was a postdoc. We took this microscope that's called a focus ion beam. So some of you saw it today. And we basically repurposed this so that we could take cells, and I'm going to show you in a minute, and carve out thin regions deep inside cells or tissue to find the thing that we want to do, like literally to try to catch the molecules in the act of behaving or misbehaving. And then eventually go to this really large microscope. So you saw the two microscopes on the right today. We have two of those. Yes, I mean, the chancellor has invested a lot in this technology. Thank you very much. Right. So this microscope, actually, when I came from Munich, is a microscope that is located here in the Qualcomm Institute. Some of you saw. This was a prototype. Once a company figured out that the hacks that we have done during my postdoc were worthy of investment, they came with a prototype that broke a lot of the things that we originally designed, but then fixed it eventually. This is the inside of the microscope that you saw today. All of this thing that you see in gold is basically something that we designed to be able to hold those two little O's, the two little rings. So that's the electron beam that I told you about. It has a scanning electron microscope and a focus ion beam. And we use the focus ion beam, which is about 5 to 9 microns, to basically raster away parts of the cell that we don't want and leave a very thin membrane that still remains sustained by the rest of the cell. And that's the part that we're going to be able to see at high resolution. So this is, again, a microscope that we just repurposed from material sciences so that we could supercool the samples inside the microscope and then literally open windows into cells. So I'm going to show you some of the data that we see, and then I'll show you how we annotate it. For those of you that might remember your biology, the thing on the right is the nucleus on the cell. That little sausage that goes through is a mitochondria. And my movie's not going to play. I'm going to try it again. No. Okay, well, you have to imagine it. It goes, there it goes. So we're going up and down. So this is 3D data. And eventually we color. So everything pink is chromatin, is where your DNA is. Green is mitochondria. And then red over there are the highways that you saw in the movie before. And those little things that look like popcorns are ribosomes. They're machines that make all the proteins inside your cell. Now, this is a cell that's a human cell. You can grow in culture. So I could take one of a little flakes from some of your skins, make them into stem cells, then give them Parkinson's disease, convert them into a dopaminergic neuron, put them on a grid, and study them for Parkinson's. So this is really fantastic. The technology that we can do it with basically every cell that we've come across so far. So I'm going to tell you only one example. There's a lot, so please ask me if I see you later. As the chancellor said, I like collaborating across many places on campus. There's 3 consortiums that we've started on campus to study many different things. This is a particularly exciting one because it's related to Parkinson's disease. About 10% of the cases in Parkinson's disease are related to a single point mutation in your DNA, and this makes you a very high risk of having not only Parkinson's, but early onset Parkinson's, like Michael J. Fox or people like that. This protein is called LRRK2. We didn't even know it existed until about 20 years ago, and then it was cloned in 2004, and when it was cloned, it means, yeehaw, now everybody wants to get the structure, everybody wants to understand it, and somehow this protein is really difficult to work with, to the point that the Michael J. Fox Foundation shipped protein to the International Space Station and taught the astronauts how to pipette to see if that way they could crystallize it and get the structure. And so this didn't work. This happened about the time that I came to UCSD 9 years ago, and a very famous National Academy member on the other side of campus told me, I just ran into this, I just learned about this protein. All the pharma companies have given up on trying to get the structure. It's impossible to purify. So I said, all right, let's go. So we tried to do a different thing. We tried to get the structure of this inside the cell without taking it out, because that looked very different. So this is a cell that we sadly gave Parkinson's disease. I particularly like this one because the Parkinson's part looks like a jellyfish. This protein made... people were a little puzzled because this protein seemed to make fibrils like other neurodegenerative diseases, but this wasn't known. When we zoomed in into this protein, here you'll see we made like this little lamella, this little thin membrane that I told you about earlier. Then we take it to the transmission electron microscopes I showed you before. And then basically we see this beautiful landscape of cells. And we label this protein fluorescently. So if you can squint a little, you'll see that the regions that are green also have some fibrils behind them. And what we discovered is that this protein is hijacking these microtubules, these highways that you saw earlier that come apart. So we thought that Parkinson's, and we're still thinking and investigating this, might actually be related to transport of proteins inside your neurons that have very long axons. So basically doing this, we focused on regions that we knew had the protein. Now we look into a lot more detail. This is a tomogram. You'll see over there, maybe you recognize them, these microtubules that you saw in the movie, but now in real life. And then basically we could sort of like say, okay, this is where the protein is, and pick out this little segment, and that way understand how the protein wraps around these things, basically blocking the way for other molecules to go through, and going a little farther and farther, always sort of zooming all the way in. So in a moment, you're going to see the structure of this protein that we got eventually. Once we had the structure of this protein, we could locate all of the mutations that are genetically present in this protein. And that actually meant that a lot of people started working on this to try to design inhibitors. And there's actually one inhibitor in clinical trials right now, which we're not involved with. We just put the structure out and people worked on it. But it was very satisfying. At that point, there's probably 8 different people on campus that are working actively on this particular protein, Parkinson's disease, looking at it inside fish, inside brains of fish, and other model systems in mice, in human brains, and also sort of like at this very basic level. And we are very lucky to have this consortium on campus. We've both been funded by the Michael J. Fox Foundation and now by the ASAP Foundation, which is sort of like a really large foundation trying to accelerate science across Parkinson. And we have, we now collaborate with clinicians. We really meet a lot of people, including patients during conferences. It's very satisfying for a physicist who grew up in Mexico to be able to be part of these wonderful efforts. I don't want to tell you other things. This is just another data from my lab that you can see. We work on this thing that's called the nuclear pore complex. We work on the blue things that are called nuclear lamina, that are the ones that will make you look like an old person if you have a single point mutation in your genome. And this is chromatin that we're also studying in the context of cancer. So we're trying to mine our data. We're now integrating new technologies. We're very excited about data sciences at UCSD because what we want to do is that we don't take advantage of every pixel that we produce here. We don't recognize some of the proteins. So we want to figure out in cell and disease in high throughput how these molecules work. And your DNA is very precious, but it doesn't make you who you are, right? And so what are the things that make you unique besides your genome is what's happening inside your cells. And what makes you tick, what makes you hurt. And we're doing this together at UCSD. This is a painting from a friend here from San Diego, David Goodsell. He made that painting without having this data, which he's very good at. And so this is what we're trying to do for all of your cells. David, actually, there was an article in the New Yorker detailing all of these efforts, which was wonderful. I just want to say this is my lab. Everything that I told you today, as opposed to the fellows, I didn't do. I have a wonderful lab of creative, bold, kind, wonderful scientists, many of them from India, some of the best, I have to say. And of course, the university has been fantastic, and we have a lot of collaborators. There's actually just a list for one of the projects. And then there's many funding agencies that have believed in our work and made everything possible. And thank you very much for having me. And it was very nice meeting you all.